Bismillahirrahmanirrahim. Assalamu alaikum students. In this lecture I am going to describe plant propagation by seed methods. This is plant propagation part 2 lecture. Let's start. As we have discussed in our last lecture, there are two types of plant propagation. The one is called sexual propagation, the another is vegetative and asexual type of propagation. Asexual or the vegetative type of propagation we have discussed in our last lecture, and in current lecture we will discuss about sexual propagation, which is occurred by seeds. Seed propagation. It is a method of sexual reproductive method in which the new plant can be produced by the reproductive part of the parent plants. The reproductive part of any plant is flower. Flower is considered the reproductive part of the plant. It is the reproductive structure in which after pollination, then fertilization, and zygote is formed by mating of two gametes, and this zygote develops into embryo in the seed. The seed can germinate into new offspring or new plant. So this is the method of sexual reproduction. By this method, plants can be multiplied. Reproduction of this method or the reproduction of plants by seeds or spores by the uniting of two gametes of spring plants are genetically different than parent plant as vegetative propagation. So variation is developed by the process of seed propagation, because offspring has half of its genes from its mother and half from its father. Offspring is genetically different from both of their parents. Like you have already studied about Mendel's law of segregation, and there are a lot of examples like red and pink flowers, red and white flowers. So variation is developed by this method. Union of the pollen, male reproductive part or male gamete is pollen, and with the egg as female gamete to produce a seed. A plant in a by this method can be produced or germinate when seeds can break their domains. Seeds are actually classified under the spermatophytes. These are the group of plants which includes angiosperm and gymnosperms. Seeds are included in all the angiospermous plants, for example in mango, in banana, in strawberries, in various other fruits, in sunflower, etc. And while in some plants seeds are naked, like in pinus and in ficus. So in all the gymnosperm, seeds are naked without fruit, while in all the angiosperm, seeds are included in fruits, because fruit is present in or developed in angiosperm while absent in gymnosperms. So in generally there are three parts of seeds. In young plants before emerging from seeds. Number one, seed coat, which protects the plant. This is the hard surface. It protects interior of the plants. The another part is called embryo. Embryo is actually the seed's food reservoir, and it is the new plant that develops as a result of fertilization. It is the plant that contain cotyledon. The upper part of the cotyledon is called hypocotyl, and the hypo is the lower part of the cotyledon, while epi is the upper part. From upper part of the cotyledon, shoot is developed, and from the lower part of the cotyledon, root is developed in the plant. So third is more important part is called endosperms. Endosperm is actually the part from where plants is a tissue produced inside the seeds of most of the flowering plants. In the plants in which double fertilization occurs, it is a triploid, meaning 3 chromosomal sets per nucleus. In most of the species, it surrounds the embryo and provides nutrition in the form of starch. So it can also contain oils and proteins. So simply, endosperms are the tissues that surrounds and nourish the embryo in the seeds of angiospermous plants or in flowering plants. In some seeds, the endosperm is completely absorbed at maturity. For example, pea and bean, and the fleshy food storing cotyledon nourish the embryo as it germinates. So you can see here the parts of seeds. So students, let's talk about the advantages of sexual reproduction and propagation in plants. First advantage of sexual reproduction is the plant raised by seeds is planted leaved. They are long lived. They are viable. They are hardy with deep root system, so they are vigorous in growth as compared to the plant which can be developed by vegetative propagation. The possibility is there to obtain change in seedling, the performance of which are better than their parents. For example, mango variety. The polyembryony phenomena of propagation of more than one seedling from a single seeds produced true to time nuclear embryonic seedling, which could be used as root stock for uniform performance. For example, mango varieties, it is very common in citrus and jamun. Seed propagation is actually necessary when vegetative propagation is unsuccessful or expensive. For example, in papaya, coconut and areca nut. Seed propagation is also have advantage when root stocks are usually raised by seeds. When seedling is required in large number, seed propagation is the only easy mean. Dry land fruit, for example, and forest species. So these are the some advantages of seed propagation. Now discuss about some disadvantages of this method, like when progenies are not true time and so they become inferior, because in the commercial orchard it is necessary to have uniform quality, growth and yielding capacity. Choice tree or any hybrid tree cannot be perpetuated true or true type by seeds except in apomixis. So the advantages of vegetative propagation, it become disadvantages of usually seed propagation. Seedling has a long juvenile period. It take more time than vegetative propagation, so it is another disadvantage. In crop like citrus, coca, rubber, the seeds must be sown afresh, and for example immediately after extraction many varieties are seedless. Seeds loses its viability in short period. So these are the advantages and disadvantages of seed propagation. Seed germination and seed propagation is actually necessary. One, vegetative propagation is unsuccessful or difficult. It is necessary for rising root stock for grafting and budding. Seed formation and maturity. Seed actually develops along with the fruit and reaches full size and maturity when fruit ripens, and hence seed should be extracted only from ripe fruit. It is in all angiosperm, seeds are enclosed in fruit, while in all gymnosperm seeds are naked because of the absence of ovary or fruit. Seeds actually gathered from immature fruit may not germinate to under favorable condition and may lose viability more quickly than full mature seeds. So only mature seeds can be germinated. Dormancy. This term is actually used to describe seeds when seed remains in slipping condition. When seeds remains in slipping condition, the term is called dormancy. That will not germinate because of any condition associated either with the seeds itself or with existing environmental factors such as temperature and moisture. Some seeds may even germinate with the fruit, for example in jack, avocado, papaya, and when seeds germinate within the fruit, the term of this type of germination is called viviparous germination. So how can seeds dormancy can be break? When seeds remain in dormant condition, several pre-germinating seed treatment can be used to break seed dormancy. For example, number one treatment is chemical or acid scarification, in which hard or important to seed coat can be break by concentrated sulfuric acid. So concentrated sulfuric acid is an effective method and is an effective ingredient to treat seed coat, soften. The purpose of this treatment is to modify hard or important or impermeable seeds covering, generally soaking seed in concentrated sulfuric acid in an effective method. The time of treatment may vary from 10 minutes to 6 hours according to different species. And after treatment, seeds are thoroughly washed in clean water to make them free of acids and then are sown immediately. That is, the seeds of cotton, asparagus, acacia are generally treated with 50% concentrated H2SO4. Thus, hard seed coat can be softened and seed dormancy can be break by this treatment. Seeds are soaked in acid for 3 to 5 minutes. Second very important treatment can be mechanical or scarification. Seeds of few species with impermeable seed coat, that is hard seed coat, can be rendered permeable to water and gases. Their germination is greatly improved by mechanical scarification in taking care that seeds should be injured, not be injured heavily. So it can be rubbed by any scrubbing material. The another method is boiled water treatment. When boiling water is poured over seeds and getting it to cool gradually for about 12 to 24 hours. By this treatment, usually seed coat becomes softened, dry and hard. Shredded seeds change into softened seeds. Thus, the seed dormancy can be break by this treatment. This treatment usually used in case of coffee plant. So this will hasten the process of germination in coffee plant. Hot water treatment, you can see here. The next very important treatment is soaking in water. Soaking in water treatment is used in many plants like gram, pea, in kishmish. The purpose of soaking seeds in water is to modify hard seed coats, to remove inhibitors, to soften seeds and to reduce the time of germination. The seeds absorb water by the process of imbibition and thus they become soft. The time of soaking seeds in cold water depends upon the hardness of the seed coat. For example, peas, bean, acacia tree. Also, you can treat coriander plants and other some plants can when they are soaked in water by imbibition, their seed coat become softened and time of germination be reduced. Another very important treatment is moist chilling or the stratification treatment. This treatment is actually well known as a simple or effective means of overcoming physiological seed dormancy and enhancing germination of many temperate tree and shrub species. This treatment actually was maximally effective after 7 days of accelerated aging, or it increasing germination from 43 to 61%. By this technique, seeds of many woody trees or shrubs are exposed to low temperature to bring about prompt and uniform germination. Stratification actually has some benefit in softening the seed coat. The seeds are arranged in alternate layers of stand in shallow boxes for pits or benches, and any treatment that is physiological chemical that weaknesses the seed. This condition helps in rapid germination, peach, jelly, plum, oats, grapes. So both of this method, boiling water treatment and chilling moist condition, both can help in the breaking of seed dormancy. So seeds can be germinated by 3 methods: apigeal germination, hypogel germination and the viviparity germination. And what is meant by germination of seeds? Germination is actually the process by which an organism grows from a seed or similar structure. The term is actually applied to the sprouting of seedlings from a seed to an angiosperms or the gymnasium. So students, this is all about the seed propagation. Seed seeds viability. A viable seed is one which is capable of germination under suitable condition, under the condition which is required to germinate any seed. The definition includes dormant but viable seeds, in which case the dormancy must be broken before viability can be measured by germination. Some seeds viability test is a rapid estimate to determine whether the seed is alive or dead. For example, that is, the embryo is potentially active or inactive, right? Dormant seeds can be viable or non-viable. If viable, its dormancy can be breaked. Through germination is the final expression of viability. A potential activity embryo with very low vigorous or a dormant viable embryo will not germinate normally. Viability means the presence of life in the seeds. Longevity means the length of time that seeds will retain their viability. Some seeds are short-lived, like citrus. Seeds are not viable for a long time. Thank you so much, students, for being with me during this lecture. I hope it is clear to you. Inshallah, we will meet in the next lecture.